Good afternoon, good morning, wherever you are. I welcome you to this second webinar between ISTA and ISF and ISTA and ISSS. So excuse that mistake. I would like to hand over to both the presidents, Julia and Steve. Julia, maybe you can start on behalf of ISSS and welcoming all the audience here.
Okay, yes, thank you very much, Andreas, for this introduction. So I won't make it long. I just would like to welcome everybody, all the members of the ISSS and then the member of ISTA for this joint seminar. I hope you're going to enjoy it. I think it's a very important topic related to climate change and changes, the phenotypic changes. So I hope in the questions we will see your interest. Thank you very much.
Thank you, Julia. Steve, up to you now. Steve, I think you're muted.
Should I start now? Or Steve, is Steve going to
Steve,
start?
I think you're okay now.
Thank you. Andreas has the power of speech over me today, and so he'd taken over, and so we have to watch him, guys. He has the power to mute us partway through, and we can't get out of it till he clicks again. So, but thank you, Andreas, and thank you, Julia, for the introduction. And like Julia said, I hope this is the second ISSS ISTA webinar, and I hope we have some more in the coming years. And I think this is a great platform for us to meet, and like it says, where science meets application. So I'm very much looking forward to listening to Eugenio and Mary Helene on behalf of both ISTA and ISSS. Thanks very much, and over to you, Andreas, to introduce Eugenio.
Yes, thank you very much to everybody. We have two panelists today here. One is Eugenio Larios and the other Marie-Helene Wagner. And I would like to thank both of them to be part of this meeting. Eugenio is, as you may have seen from the announcement, from the Department of Ecology, Evolution and Marine Biology of the USC, University of California, and Santa Barbara. He's got a doctoral degree as a postdoctor. I think you're still there, Doctor of Philosophy, Ecology, and Evolutionary Biology. I hope that is still correct. And I would like to hand over to you to tell us a little bit more about the topic. And I think your presentation is about genotype, bioenvironment interactions in seed size are ubiquitous in relation to water availability, evolutionary potential through phenotyping plasticity, which is a quite long one. And You don't have to read it again, otherwise your whole 25 minutes are gone. Continue, just start, and Marie-Helene and I will close our cameras, and it's all up to you now.
Thank you very much for inviting me. And yes, the title was too long, so I changed it to The Evolutionary Potential of Seed Size Through Phenotypic Plasticity of a Desert Annual Plant. I am Eugenio Larios, and today I'm going to talk about a work that I did with Dr. Susan Mazer from the University of California in Santa Barbara, with a postdoctoral fellowship, UC Mexes. And yes, let me just. A central interest in evolutionary biology is to determine the amount of genetic variation and the genetic structure of fitness-related traits of wild populations. In order to understand their evolutionary potential and their adaptive nature, for instance, the ability for wild populations to adapt to a rapidly changing environment depends on genetic variation on fitness-related traits, such as seed size. For plants, seed size is a functional trait with important fitness consequences throughout their life cycle. Larger seeds are better provisioned than smaller seeds to cope with stressful conditions that plant populations face in the wild, because they are able to produce larger seedlings that are better able to reach moisture or light than smaller seedlings. Seed size has been shown principally to influence early life history traits such as germination percentage and speed. Although there's not a clear consensus on whether larger seeds germinate more readily than smaller seeds, this relationship seems to be species and environment dependent. Seed size also influences seedling survival and establishment. This figure I'm showing here shows that seed size is positively correlated to lifetime survival and negatively correlated to within-year variation in lifetime survival in a geographic gradient in the Mediterranean region, suggesting the positive effect of seed provisioning of larger seeds on fitness. Let's see. But seed size is not only good for early life stages. There's also evidence that seed size extends to adult stages such as fecundity. In this study, we demonstrated that plants originated from larger seeds tended to be more fecund, so they produced more seeds in adulthood. In summary, from the offspring point of view, seed size seems to be under selection toward larger seeds. However, the evolutionary potential of seed size is limited by its low levels of heritable variation. The evolutionary response to selection, delta Z, in this equation here, depends on both the relationship between the seed size and fitness, which determines the selection gradient S in this equation, and the amount of additive genetic variance described here by narrow sense heritability, or little h squared. Seed size is a quantitative trait, or a trait that is influenced by multiple genes or alleles at multiple loci. Each of these alleles contributes a small fraction of the phenotype described by total phenotypic variance. Additive genetic variants are those genetic effects that contribute to the phenotype in the same way in every genetic background. So they are genetic effects transmitted by the nucleus in recombination that we can reliably predict from one generation to the other. Quantitative traits such as seed size can be described by the normal probability distribution. In fact, the more loci that contribute to a trait, the more normally distributed the phenotype will be. Because seed size is a result of sexual reproduction, total phenotypic variance in seed size is influenced by maternally and paternally inherited genes. However, the maternally inherited genes are greater in effect and usually greater in effect, at least in seed size, and more varied than paternally inherited genes. We can see that the paternal influences are only exerted by those genes expressed by a fertilized ovule through the pollen, while maternally inherited genes, besides those genes expressed by fertilized ovule, there's also cytoplasmic genes and the influence of the maternal sporophyte genes that determine seed provisioning. So if we can isolate the additive effect of paternally inherited genes, given that they are from a single origin on the mean seed size, then we can estimate additive genetic variance or narrow sense heritability of a trait or their evolutionary potential. But there's other sources of genetic variation, and those come from interactions between all these additive effects. Those are dominance, epistasis, the interaction between cytoplasmic and nuclear genes, and the interactions between maternal and sporophyte and the embryo. Dominance refers to those interactions between alleles in the same locus, so alleles that influence individual seed size. Epistasis refers to an interaction between alleles but at different loci. All genes or interactions between genes that remember that the quantitative traits are
Could you open your microphone up again?
Oh yes, sorry, I'm sorry.
May I ask all participants to stay muted if you're not speaking? Thank you very much.
Yes, we can detect them when reciprocal cross don't have the same seed size. For example, individual seed size from plant A as a mom and plant B as a dad don't have the same seed size as plant A as a dad or plant B as a mom. And finally, the maternal sporophyte and the embryo interaction can also influence seed size. And statistically speaking, all these interactions between paternal and maternal genotypes can be due to all of these interactions, and they are really hard actually to tease apart or separate. Putting these all together, we have that all these genetic factors are part of total phenotypic variance, which is the actual expression of the phenotype in the wild, I mean in the plant, and they are expressed by this sigma p square. Actually, total phenotypic variance is composed of genetic variance and environmental variance. Genetic variance it's also composed from additive and non-additive components. And the additive are those paternally and in maternally inherited genes, the effects of those, and the non-additive are the interactions. So what does genetic variation has to do with heritability? So heritability is the proportion of total phenotypic variance that is due to genetic variation, and it comes in two flavors: the narrow sense heritability and the broad sense heritability. Narrow sense heritability is just the proportion of additive genetic variance to total phenotypic variance, while the broad sense includes the non-additive sources. This thing. So an additional kind of worms, it's the effect of the environment, and that's where plasticity comes into play. So maternal environmental effects, for example, are those environmental conditions experienced by the mother plant during seed provisioning, and they affect seed size. For example, the amount of competition, the amount of water availability, or light, or even temperature. So there's the error variance, also a source of environmental variation that is due to non-explained sources of variation. Some background on the motivation of studying the quantitative genetics of seed size came from the results of a previous study where we used a winter annual plant from the Sonoran Desert to investigate the effect of competition and seed provisioning and its consequences for seed dispersal in an attempt to study the relationship between seed size and seed dispersal. Seed size and seed dispersal are theorized to co-evolve because they represent common solutions for shared population dynamic functions of seeds, which are the reduced risk in variable environments, or they do through bed hedging, and the negative effects of crowding and seed competition. In this study, we hypothesized that the trade-off between competitive ability and seed dispersal would be related to the escape of from crowded conditions for a successful establishment. We knew that larger seeds had higher competitive ability in another study, and if smaller seeds were able to disperse farther, there was a potential for a competition dispersal trade-off in this species. We found that plant density caused maternal plants to reduce offspring provisioning. This was an observational study, and that smaller seeds were able to disperse farther away from their mothers. Furthermore, with this winter annual, we were able to estimate heritability of seed size through a parent-offspring regression, and found that maternal effects were responsible for the reduction in offspring size rather than a genetic effect. And we can see this in this path analysis that the relationship between maternal and offspring seed size is through plant mass. And so this suggested that phenotypic plasticity was responsible for seed size, the seed size of the offspring. So we were able to perform these parent-offspring regressions because our study system can tell us about the size of the maternal seed in retrospect. Diphtheria californica, it's a winter annual plant from the mustard family that has a persistent seed ring that stays attached to the root even after they have grown and reproduced. A serendipitous feature that has allowed us to study natural selection on seed size in the wild. This is a picture of the persistent seed ring attached to an adult plant of Diphtheria californica. So we measured this and related to fitness and to study natural selection. So in this study, we used a quantitative genetic approach to reveal the genetic basis of seed size and the potential plastic response to water availability in the winter annual plant, Diphtheria californica. And we asked the following questions: what is the evolutionary potential of seed size in Diphtheria californica, or what is is there any narrow sense heritability in seed size in Diphtheria californica? Does seed size evolve through phenotypic plasticity or just additive genetic variance? And what is the effect of water availability on seeds on the phenotype? So to do that, we used a diallel design, mating design, which is a quantitative genetic procedure that involves crosses between 5 individuals and the reciprocals. And we excluded self-pollinations to generate pedigreed seeds of 20 genotypes per diallel. We replicated 3 diallels per population, each one replicated at the same, each replicated in wet and dry environments to be able to estimate genotype by environment interactions. These 3 populations belong to a geographic gradient in North American deserts, Mojave and Sonoran deserts. With these, we estimated variance components of seed size, or the genetic the genetic components. In this case, with the dialyls, we were able to estimate the general combining ability, the specific combining ability, and the reciprocal maternal effects, which are reciprocals of those combining abilities. And this is more or less the cartoon of the experimental design. We excluded cells here, but made it each of the 5 plants reciprocally. And yes, and so we initially in 2017 collected seeds from at least 20 maternal lines in 4 populations. We after ripened those seeds until the spring of 2019, where I got the postdoc fellowship, and germinated them in the greenhouse, in pots in the greenhouse at the University of California in Santa Barbara. We didn't have a germination of 1 population, so we were left with 3 populations and 3 dialyls for Kelso, 1 dialyl for Lobos, each one replicated in a wet and dry environment, and 3 dialyls for Seri population. So we have 7 dialyls in total. Why it doesn't? So this is a map of the source population. The Kelso dunes are in the Mojave Desert here in California, and there's 2 populations in Sonora, in the coast of Sonora, in coastal dunes. And initially we had another one in Pinacate, but didn't germinate. So this is the flower. Each of these flowers of Diphtheria californica produces a pair of mericarps that we produced by hand pollinations. In order to do the hand pollinations, we emasculated the flowers, and in this case, it's easy to do with only cutting the tip of the flower because the stamens are longer than the style, the stigma, and so it was an easy task. And this is what they look emasculated. So we pollinated those stigmas manually. We have a wonderful help from UCSB undergrads. Otherwise, it wouldn't have been possible to do. So we produced pedigreed seeds that we color-coded when they were in the plant, so we could know their pedigree. One of the colors is for the mother, and one of the colors is for the father. It was that was a fun thing. So with all these pedigreed seeds, we analyzed them by photographing the seeds per genotype, and using the ImageJ analysis software, we measured 2 axes of seed diameter to estimate an ellipse area in millimeters, in square millimeters. So we, at the end, produced more than 8,000 seeds with known parentage. The statistical analysis was used so we did the random diallel analysis, and this long equation, it's describes all the variance components that you can do with this specific diallel design without it's a complete diallel without selves. So we have the mean seed size, overall mean, the general combining abilities of the ith and the jth parents, the specific combining abilities of the cross of the ith and jth parents, and the reciprocals, and B is the water effect, and this is the error. So we had a seed size as a response variable in terms of ellipse area, as a function of water, which was our fixed effect, and all the random components variance components were random variables. Those are GCA and GCA by water interaction, SCA and SCA by water interactions, and the reciprocals all tested with a water interaction. So these water interactions would tell us whether there is genotype by environment interactions. So just some cartoons of what the GCA, the variance components are. So the general combining ability is the average performance of a genotype in a hybrid combination. So for example, father one crossed against all mothers in a hybrid combination, and in the same mother one crossed against all fathers again, and relative to the overall mean. So this is the additive effect of the genes or the breeding value. Specific combining ability refers to phenotype deviations of specific parental combinations in relation to average parental performance. Now we're not relating, so we're not doing the hybrid combination, but just a specific combination in relation to their parental mean. So this refers to non-additive effects of genes, such as interactions and dominance, epistasis, and so on. The reciprocal general combining ability is the variance due to effects of the individual functional gender on offspring genotype. So this is similar. It's just that the reciprocal of the GCA, but it's not relative to the grand mean, but to the parental mean. And the reciprocal specific combining ability is variance. It refers to variance due to the effect of cross direction on offspring genotype. So in our first the specific combining ability, it didn't matter the direction of the cross, but here this is what we're testing, whether father to I mean if we father two, mother one, mother one, and mother two. It's different to mother one, mother two, father one. So the reciprocal. And so the results are that. So I'm going to guide you through this table. I'm sorry if it's too messy, but so these columns here refer to the populations. So the Seri, the Kelso, and the Lobos population. The first column are the variance components, the variance components and their water interactions, and the residual. So these are variance in this is the first number and their corresponding standard error. Bold letters correspond to statistically significant effects. And as we can see, GCA, so the general combining ability by water interactions is consistently significant in all populations, meaning that there is, well, this all too means that there is genetic variation for phenotypic plasticity or the potential to evolve through phenotypic plasticity rather than genetic variation alone. We can see that GCA is in some instances is 0, but it's non-significant. So there's no additive genetic variance for seed size in this population, as predicted actually by evolutionary theory. We also have that the reciprocal specific combining ability is consistently significant in all populations, meaning that a maternal effect, like a non-additive maternal effect, is also playing as a phenotypic plasticity. And finally, we have the norms of reactions that we estimated from the residuals of the ellipse areas. This is in relation to the dams or the females. So we subtracted, in order to estimate the residuals here, we subtracted the mean of the dams per dialect. And we can see, what we can see here is that so the K and the S populations have similar patterns, where the variance of seed size across the genotypes doesn't vary much, but what varies is the rank of which is the largest seed size in a dry in a wet environment. Same here in the S, whether while in the L population, what the variance does vary in between the dry and the wet treatments. We can see the variance here is smaller than the variance in the wet environment. And this is with regards to the females. We can just look at the y-axis is now well, the variation in the y-axis compared to the next graph, that is the norms of reaction from the sire perspective. So now the residuals are estimated with respect to the mean of the sires, mean seed size of the sires produced by sires, and we can see that there's the same pattern but just less variation overall. So we can get from here that the dam influence on seed size is greater overall than the sire influences, as expected, and so the same patterns arise within the figure. I mean, this only changes. There's a slight change in variance here, which is opposite in both directions in both populations, but the greatest change is in the L populations. I have to say that the L population, since there was only one pair of dialyls, but yeah, I don't know. This population behaved kind of strange, making really big and really small seeds. So in conclusion, we can see that there is genetic variation for phenotypic plasticity in seed size, so that seed size would evolve through changes in water availability. At least that's what we tested, and we had some evidence before from the amount of competition. Narrow sense heritability is close to zero, as expected from evolutionary theory, because so traits that are fitness-related traits that are in that have a natural selection acting very strongly on them tend to be to have no narrow sense heritability, because the action of selection erases that additive genetic variance. So we also can conclude that environmental heterogeneity, if could maintain genetic variation in because phenotypic class, there's no a predictable plastic effect on water availability on seed size, but some genotypes make smaller seeds under wet environments, and some make big, and you cannot tell whether there's a homogeneous prediction, homogeneous effect, so it's unpredictable. And with that, I would like to acknowledge the UCMEX's postdoctoral fellowship, the CONACYT, the Maser Lab, and all the amazing undergrads that helped me do this. Thank you.
Thank you very much, Eugenio, on that. I got a question during the presentation, not related to your presentation, but how to enlarge the presentation. There are several possibilities. You can go on the presentation, scroll in, scroll out. Above the presentation, you can have a drop and drag menu, which you can make bigger, or to zoom above in the above menu. You can also zoom it up in there. So if you want to have it a little bit bigger, or to zoom in and out, you can go on the presentation directly and zoom in and out. If you have any questions, we are happy to answer them at the end of both sessions. And please put your questions in the question and answer box, and we will try to address it at the end of the sessions. Thank you very much, Eugenio, for this presentation. We will have probably questions to you a little bit later. I would now kindly ask our next presenter, which is Marie-Hélène Wagner. She is currently working at the Gervais, and currently means for quite some time in her career, the Station Nationale d'Essai des Semences in France, and her presentation is about seed germination phenotyping and controlled conditions to measure genetic diversity and address climate change. Marie-Hélène is an engineer, and she studied plant physiology at the University of Paris. And you're also active in other areas like biostimulants and plant bioprotection, and we are very happy to receive your presentation now, Marie-Hélène. Thank you for being here with us today.
Okay, thank you, Andreas. So I'm going to share my screen. Is it okay for you?
It looks good. Thank you very much.
Okay, so in full screen, maybe it will be better. So hello, everybody. I'm very pleased to present our work in which is in the connection between the two associations dedicated to seeds, so ISSS and ISTA. And I've been working in the germination lab led by Sylvie Ducournau for now 27 years without the same patient at the beginning. And so you won't be surprised that my talk will be on seed germination phenotyping. This phenotyping has been improved in the last decade thanks to image analysis. And so we work a lot with Didier Demilly, who is in charge in GEVES group of image analysis phenotyping, applied to seeds, but also to varieties, the other part of the group studies. And the tools I will present now are part of larger services offered by a platform in Angers, in the west part of France, called Phenotypic. So my talk is divided in 3 parts. I hope you don't see the screen on the right, which is go to webinar. Can you confirm that you see only my presentation?
Yes, we can confirm that.
Okay, because I've got another panel. So to avoid losing you, 3 main points. First, I will present the tools and mostly the data that we can obtain thanks to image analysis. I have selected 3 examples. The pictures on the right part of the flowers may give you a clue to find which species I'm going to talk about. And I will discuss about the continuous improvement, about methods to phenotype early stages of plant life. In control conditions, it can take 1 week, because we control conditions and we often use favorable conditions. But our aim is to have an idea of plant emergence in real life, that is to say in field or greenhouses or in gardens for some species. And the timing in this part can be spread larger than, of course, in control conditions. So the tools themselves were developed a long time ago with the first prototype in the 19th. And now we've got 4, I have to change this window because I don't see my screen, okay. So in the pictures, you see that the tools were developed upon incubators, which are Copenhagen incubators or Jacobsen tables, which allow thermal regulation at plus or minus 0.5 degrees, because the regulation is done with water. Upon this tank, 4 cameras per tool have been put, and all the system is monitored with a software in order to take images at a determined frequency that we select, combining temperature and species. Now we've got 40 species which have been validated for seed imaging with a quite high throughput. mainly agricultural, sorry, I didn't put my pointer, mainly agricultural crops. You can see on the diagram with oil crops, but also beet, forage, but also vegetable crops and model species. Model species because when we began, the idea was to develop tools for seed physiologists and to increase the throughput of germination phenotyping when genotyping reached very high throughputs. The second application, of course, is seed testing. I'm working in the official seed testing laboratory in France. And this tool can be used for seed vigor testing, but also to check seed technologies improvements. Andreas has spoken about biostimulants, but before that, priming treatments or other seed treatments, we can measure the impact on seed germination using image analysis. And in the last years, some breeders came to us to screen their genetic resources. Regarding the data that we obtain with our tools, I'm sorry for the seed physiologists who are in the audience. They already know what we obtain. We obtain mainly curves, of course, because a germination progress curve is obtained having the percentage of germination according to time. So here, 3 examples, 3 seed lot of Medicago truncatula, which is the favorite species of Julia. And yes, we've got the curve. I pay your attention on the number of experimental points. The frequency here is 2 hours at 15 degrees. So we get a lot of points, which avoid to fit with a sigmoid model called Gompertz. But you can see that experimental data and computed data are very close. And the main traits that I will use during the presentation are represented by this line, which represents T50, time to reach 50% of germination. The 3 samples have different time. The most rapid 41 hours to complete 50% of germination, and the orange one 70 hours. So the germination rate is very different between the 3 samples. But pay your attention that the total germination after 5 days at 15 degrees is also very different. The blue one germinates fully, as the orange one only 70%. Thanks to image analysis, we have more than germination curve, because here you've got images of Medicago all along the time. And before germination, we can access to imbibition rate using evolution of seed size upon time. And after germination, in this part of the curve, you can see here the radicle begins to grow. Of course, it depends on the throughput, on the number of seeds that we will put on images. And there's a special image at sowing time here at 0 time. We can use, so it's a connection with Eugenio presentation. We can have a good proxy of seed mass. These are results on all seed rape, where 4 cultivars, 2 hybrids and 2 lines were analyzed on 3 different grading after calibration in 3 sizes for each varieties. And we obtain a good relationship by weighing seeds, single seeds, and seed volume obtained on this first image. The last parameter I would like to introduce is to score germination rate is mean germination time, because image analysis is done seed per seed. And so each single seed is scored for its germination time, and pulling all together the different replicates, we obtain mean germination time. In this example, T50 and MGT are very close because we have good germination samples, because we are in good at a favorable temperature. Regarding temperature, I thank here Takashi Shinohara and ISTA, who has funded a special project dedicated to Brassica species. And this is an example on black mustard, which species we had three different cultivars, varieties or genotypes, if you prefer. One here called C1 in blue with 4 seed lots. Here, a yellow variety of black mustard, which is strange. So seed coat has been modified, and we've got we received 2 seed lots for this orange variety, and the green one, C3, 4 seed lots. At 25 degrees, you can see that all 10 samples have the same shape of a germination progress curve. In 4 days, all are finished to germinate, except this green one, which is, I think, a C32. So the mean germination time range is half a day, so 13 hours at 25 degrees. When we decrease with a range of germination, which is quite important, but due to only this seed sample. At 20 degrees, we've got a second green seed lot, which is delayed in germination and which has not finished to complete germination after 6 days at 20 degrees. The other one, the range of germination rate increased, and we've got 38 hours of difference between the most rapid, here the blue one, and the last green. At 15 degrees, the interesting thing is that the 4 seed lots of the first variety still germinate fully in 7 days and have the same germination rate. So this variety is tolerant to decreasing temperature, and for sure there's a genetic effect. As for the other one, we've got also a seed lot effect, or a seed production condition effect on the germination rate and on the final germination after 7 days at 15 degrees. Here, the germination rate range is very large, 61 hours, and we've got circa 50% of differences in total germination. So now, if you didn't guess the examples, here you've got the seeds of sunflower, oilseed rape, and Medicago truncatula. I have selected them because they are related to climate change. The two oil species, because well, for sunflower, the strategy to avoid drought and high temperature during flowering is to sow seeds early in the spring. So early sowing means cold temperature in spring, at least in Europe, to escape dry weather during flowering. For oilseed rape, especially winter oilseed rape, which are mainly used in Europe, they are sown in summer. And so the strategy is to screen adapted genetic resources in case of, and it becomes more often now, in case of dry conditions when sowing at the end of summer. And the last example, I thank warmly Julia Butank, who allows us to use the data we provide her and her team in one of her research project. She has coordinated a few years ago called Reguleg. which aims to analyze regulation of in legume species to find, to use the plasticity of germination and to measure the impact of climate change in this plasticity. So we will see that at the end of the talk. Regarding the methods, so here you've got always the same pictures as before. Two automated tools. One is is lightning to take a picture, and here is the system for sowing. So sowing plates where we can put from 200 seeds for large species like sunflower here until 600 seeds per camera in case of small seed as Medicago or oilseed rape. For each species, so imaging is done on top of paper, special blue thick paper. And we often use replicates of 25 seeds per camera, and we can mix a sample under a camera. So here you've got an example, by instance, for sunflower of 8 different genotypes, each on 25 individuals, and we repeat under each camera in order to get at least 100 seeds at the total. I have pulled here in favorable condition for the 3 species, so 25 degrees for sunflower, the most sample we have analyzed. So in the case of sunflower, 100 samples. And here you've got in blue the mean curves that we can obtain and the diversity. And you see that at 25 degrees, in 5 days, all samples have completed germination. In green, it's a virtual sample where all the rapid seeds of all this material, so 100 samples, are pulled together, and you can see it's the upper limit ideal curve for a perfect seed sample, which doesn't exist in real life because it's not made in a factory, but in field. And at the opposite, in red, the slowest ones are pulled together, but you can see that in sunflower, we can reach 70% of germination in these good conditions. For oilseed rape, we have analyzed more than 1,000 samples at 20 degrees. 20 degrees is very favorable for oilseed rape, and in 3 days, germination is finished. But the lowest quality virtual seed lot is lower and reach only 50% of germination. So we begin to have seeds which don't germinate in this timing at 20 degrees. For Medicago, we've got more than 300 samples analyzed at 15 degrees. Again, the blue shape with the diversity. And in 5 days, the germination is finished. For the red curve, the shape is very different from classical germination curves, but it could be explained, and we will see it later, by dormancy in this species. So the first example is sunflower. And we were asked to analyze cold tolerance in a panel of genetic resources of 2 breeders. And their question was, is there an impact of oil composition in tolerance to cold temperature? And we begin at 10 degrees. At this period, we can guarantee 10 degrees plus or minus 0.5 with our tools. And this diagram answer to the question. In yellow, you've got the oleic material, so enriched in omega-9, so the modern varieties that were developed at this period. and their mean germination time distribution is very similar to the more classical genotypes, linoleic, so enriched in omega-6 fatty acids. And you can see that, yes, the two graphs are very similar. So the answer was no. The composition in fatty acid doesn't impact the germination in cold temperature. But between tolerant to 10 degrees for germination and susceptible genotypes, we can have a difference of 2 days for the T50 between the two mean curves of several genotypes, with a big increase of variability for the sensitive one, because 10 degrees begins to be a suboptimal temperature during 10 days at the total. The second very important result that we obtained in this project was that germination and growth don't have the same requirement for temperature. And on extreme, contrasted genotype, 1 hybrid and 4 lines, female and male, with different oil composition, we phenotype deeply this material for both germination and growth at several temperatures. And the main result, so you've got all data, so there is diversity between genotypes for both stages, but the main result is that for germination, germination is possible from 2 degrees Celsius in mean, but for seedling growth, you need 4 degrees more to achieve growth. So after germination, you have you need a higher temperature to allow the seedlings continue its life after germination. Especially this line, linoleic, is even 8 degrees. So that's why, but, oh, sorry, to have a more discriminant test between genetic resources, we decrease the temperature, of course, only for the germination part. And by this period, the tools were improved for regulation, thermal regulation, and we could decrease two new tools were in a climatic chamber, and we can decrease temperature until 5 degrees to come near the base temperature, but not at the base temperature. During 14 days. So here I have represented the mean germination time courses for 119 samples composed by hybrids and also parental lines of these hybrids. And yes, we will reach quite a high germination. Sunflower can tolerate a low temperature for germination, except, of course, for the lowest, the low vigor seeds which compose the samples, where germination, this temperature, 5 degrees, begins to be lethal for some seeds. These are the row curves, and between I pay your attention that we lose a lot of mainly lines which don't germinate after 14 days. So T50 can't be calculated for them, but for the rest, we've got 1 week of difference between the most rapid and the slowest one, just to reach T50, and germination begins only after 4 days, so 96 hours. So the important here is to have a very accurate temperature to see these differences. To finish with this example, I have put here, I'm sorry, it's in French, but I like the drawing. It's an advice for farmers which insist on a rapid emergence. So many advices regarding soil conditions to get a rapid emergence. This rapid emergence is needed to, I translate the sentence, to reduce the period when seeds and seedlings are exposed to birds or other biotic stress. And they recommend to select an adapted variety. So according to your to the place where you are, at least in France, you have to select to check the precocity of the variety. But here in our example, I have selected 2 hybrids produced the same year, but in 8 different environments. And you can see the germination time courses obtained at 5 degrees. Hybrid 1, we've got also 3 seed lots more, which were older, but with 11 seed lots, all germinate in 14 days at 5 degrees, with a quite straight difference between the most rapid and the slowest one. But it's not the case with this hybrid 2, where we've got a big difference between this one, which is very similar to hybrid 1, and the other one. So the cold tolerance will depend on both genetic part, but also on the conditions where seeds were produced. So yes, choose the good varieties, but also a good seed lot to be sure, especially if you are in the north of France, to establish a sunflower crop. So stop with cold. We turn now for with well, not winter oilseed rape, but as I previously said, it's sown in summer. So the first results we obtained in an international European project between Germany and France called ComVigor. It aims to phenotype vigor of winter oilseed rape. And the first result was to, after having phenotyped a collection of different varieties, more than 200 varieties different produced in 2011 and 2012 for the project by partners. And we phenotype at 20 degrees the germination of all this material. And the first result was to conclude that breeding has changed, has improved varieties of winter oilseed rape by eliminating erucic acid and glucosinolate, 00 varieties, or canola in some other parts of the world. And this variety germinates slower than the former ones, which contained these 2 molecules. So by improving grain quality, we had an impact on seed quality. Between all these varieties, 12 were selected at 20 degrees to be analyzed, to be phenotyped in field trials. Here you've got the 12 curves for seed lot 1 at 20 degrees, and below the 12 curve. So the second seed lot produced in Germany, the first one was produced in France, had a higher quality, especially due to only one variety, where the first seed lot was delayed and poor germination. But in mean, we've got only 3 hours of difference between the 2 seed lots, and the germination was high for both seed lots for all this material. Interestingly, at the same period, within the Vigor Committee of ISTA, in collaboration with Alison Powell and Stan Matthews, we produced data on oilseed rape on 9 seed lots from the same, having the same genetic background, the same variety, but 9 seed lots. We to calculate MGT, Stan previously demonstrated that mean germination time was correlated to field emergence in maize. But to get mean germination time, you need the whole curve, which is time consuming. And we can summarize the full curve and MGT by an early count. So in the case of oilseed rape, it's 30 hours after sowing, a count of radicle emergence. So an early count of germination, is a good proxy of mean germination time, and we obtain a good relationship between this radicle emergence in percentage, so a very early count, with field emergence in UK. In Convigor project, so extreme genotypes at the total 36 with the variety collection, but also elite breeders material, 2 seed lots for each produced during the project. On this contrasted genotype, which were sent to field, we will see later on, we did a larger phenotyping of germination, changing the temperature, but also changing the conditions of water availability. And I have extracted here an example of 4 varieties, which in good conditions with no water stress at 0 megapascal, the 4 varieties germinate at 95%, so no differences in mean germination, but we can measure already differences in germination rate, here expressed in day -1. So too rapid, the green and the brown, very rapid germination, and the blue one, a delayed one. In the same conditions, favorable at 20 degrees and with water. When we decrease the water availability, of course, the general germination rate decrease also. The germination remains high for 3 cultivars, except the gray one, which is not the slowest one, but which germinate only at 55%. And again, the green variety is rapid, followed by the brown and the gray. When you analyze with a water stress, here only the green one is capable to germinate quite rapidly, but threefold less than with water, and germinate fully. The slowest variety, it's interesting, it's the slowest variety, so sensitive to water stress, but is capable to germinate until 80%. It's not the case of this one, the gray one, 30% only of germination. These extreme genotypes were sown in field 2 years and they show high differences also. 9 locations were used, 3 in France and 6 in Germany. But the early sowing conditions were difficult to reproduce. The idea was to mimic a drought in real life, so in field. And I have pulled here the results, the mean results of the 36 genotypes in the 9 places. In the north of Germany, it was repeatable between the 2 years with an early sowing, but it was not the case again in Germany, in Seligenstadt, by instance. In 2012, only 11% of field emergence, which is very poor. And a year after, with the same early sowing date, but of course climatic conditions, very different climatic conditions, a very good emergence, 87. So faced to this, well, time and space consuming trials, and because of the diversity of stresses, abiotic, drought, temperature, and many other, and also biotic stresses. On 5 cultivars, we use the deep phenotyping for both germination and growth, using also climatic and soil data inputs. Caroline Dürr used a model called SIMPLE for simulation plant emergence, which was validated on these cultivars through field trials in 2014. So a first data set with real experimental results in field to validate the model. And she carried out virtual sowing, I will call them like this. So more than 1,000 using 4 sowing dates, 2 locations during the period 1985 and 2012, where she got climatic data. And this is one of the table of the paper, which was published comparing field emergence according the year of sowing for 4 dates of sowing, early sowing at mid-August in 2 places in Germany and France. The early sowing is where you've got, so we've got 5 cultivars, is where you have the most difference between years. And some years are very dry already in the past, and the field emergence is very poor. And when you go for late sowing, so the end of September, sorry, in Germany, you see that the difference between years is lower, and you've got, yes, around 80%, well, 75% of emergence, whatever the year. It's not the case in Brittany, in France. Some years can be very dramatic for field emergence. And especially here, it is 2012 for both sowing, and we obtain, of course, in early sowing for more cultivars, only 45% of emergence, which is consistent with simulation, virtual sowing. So it's time now to go to the third example. And the question is now, what happens to seed traits, seed germination traits, when mother plants are grown under water stress conditions? So this time it's Medicago truncatula, and the data were provided to Julia Butank for her project, which aims to identify regulators of legume seed adaptation to environmental changes. For that, her team has produced 200 ecotypes in the seeds of 200 ecotypes, which were also sequenced for the genome-wide association studies. And they produce the seed in 2 different conditions, one with a well watering and the second one with water stress imposed just after when flowering begins in the greenhouses. So of course the yield for water stress treatment was less important than in the control situation, and for some accession no seed were obtained for phenotyping. So at the total we received 177 accession, which are comparable in the 2 situations. Using a relationship, I have previously shown the relationship in oilseed rape. This is the relationship in Medicago truncatula between seed mass, really weight in a balance, and the seed area measured in image 0 or after the beginning of imbibition. This relationship was interesting, so I used the dry seed area obtained on image 0 for the phenotyping of this material. And you can see in yellow the distribution for water deficit seed production, and in blue the control conditions, and the water deficit conditions provide smaller seed for the water stress plants give smaller seed compared to the control plants. And our colleagues have also weighed the seeds, and these seeds are also lighter. So it confirms that there is a relationship between seed mass and seed area. Here is the germination. So the main result is that seed produced with water stress germinate faster than control seeds at 15 degrees. You've got here the mean parameters and the range of each parameter. Here you can see 4 hours of differences for circa 200 accession of difference, with a range very comparable between the two treatments, the same for T50. And the beginning of germination is only 2 hours in advance for the water stress samples. I have extracted 3 examples. Genotype 20, which reach 84% of germination in the control condition. So it's the minimum germination obtained in the phenotyping. And you can see that the orange curve, which represents the germination of water deficit production seeds, which germinate faster than the blue one, the control, and 14 hours of difference in T50. A second genotype, 42, ecotype 42, which fully germinate in both conditions of production, with a light advance for the wild deficit seeds, 7 hours in advance in T50. The last example is very interesting because it was the main difference in the mean parameter. Is the mean germination obtained after water stress is 30%. This is the curve. And this ecotype, going back to the images, we can see that we have hard seeds there, and there. So impermeable seed coat, and also fresh seeds. So seeds were imbibed but didn't germinate. That's why. So this ecotype produced in water deficit condition produce dormant seeds mainly. So all these data, Julia told me that she was very interested on this data. So they are valuable to continue for her study for GWAS on germination plasticity. And for this reason, that germination rate depends on genotype, but also on environment of seed production and on the interaction of both. This is the T50 distribution of the two environment, control in blue and water deficit in yellow, so in orange. I prefer this beautiful violin diagram that Julia produced and allow me to present you. You've got again in blue the control conditions, and all the ecotypes are distributed into beautiful violins, with wild deficit having a lower T50, but a spread larger than in control conditions. And the interesting graph for GWAS further analysis is that if there are some genotypes, the genetic effect is significant in all situations, but in gray, the genotypes have the same behavior when they are produced in control water conditions or in water deficit. And in green and red, you've got significant interaction. In green, the example like ecotype 105, which germinate faster when produced with water than and which is dormant when produced without enough water. And the most ecotypes has a red response. so they germinate faster when produced in a stress condition compared to the control. Well, but the end of the story will be told by Julia in further publications. So to sum up my presentation, I would like to insist that on germination is just the first stage of plant development, but it's a main stage. It is necessary, not sufficient, but necessary. And thanks to image analysis development, now we've got accurate and reproducible germination curves. That's why we can use germination rate better than only, well, total germination, where the range is less important than for germination rate. We've got also access to other parameters, seed size, by instance, thanks to single seed traits analysis, and also imbibition and the beginning of radical growth. And germination is necessary for emergence, but not sufficient. We need also the other part, the seedling growth. For this, we have developed another kind of tool with a lower throughput, but it's again, we can control the climatic conditions. We can study 400 seedlings at the same time using 2 cameras, here and here. And the idea is to mimic darkness for seeds. That's why you can see that the lighting is done in green conditions in order to, yes, to mimic what happens in field or in greenhouse until seedling has emerged. We would like to improve our tools to study the water stress studies, because so we imagine that we can improve image analysis using plastic boxes and improve lighting system to avoid to have a reflection of light on plastic boxes. Plastic boxes is also important to work on seed microbiome, which is a new project which has begun in Angers. And we will analyze seeds with their microorganism, and especially for bacteria, our system which regulate temperature with water could be a limit when we study microorganisms in these conditions. So we will improve using closed boxes. I would like to thank a lot of people. Some names are listed there, mainly colleagues from the Germination Lab. So here, this is France, and the Germination Lab is located as GEVES headquarters in Angers, in the west part of the country. But I thank also many colleagues from INRAE, also scientists from universities in Paris, Aberdeen, and Tokyo, and also colleagues from private seed companies. The collaborative project can't exist without funding, so the National Agency for Research has funded 2 of the projects I have mentioned, but also a private support of seed companies for research on oil crops, French support, and also ISTA for the funding the ISTA-ISSS special project. And thank you for your attention, and I wish you the best for the end of the year.
Thank you very much, Marie-Hélène. We overrun the whole time now by 25 minutes, and we are losing a number of attendees in the moment. I think I thank everybody of you. I will not go through the questions today. They are recorded, and you should have received them Marie-Hélène and Eugenio, maybe you can try to answer them and bring them up at the next time, or to someone else. I would like to thank everybody for being here, and all the interested parties. Maybe Steve or Julia, you would say some final words.
So, like you said, Andreas, it's been a very interesting presentation today, and I much appreciate the time from Eugenio and Marie-Hélène, especially summarizing a great range of projects across many years of work. So I look forward to updates in the future from Eugenio on his project. Was very interesting about the genetics and how that interacted, and also updates in the future. So thank you very much to both of you, and I much appreciate your contribution to the ISTA-ISSS seminar today. So maybe over to you for the last word.
Yes, well, I would like to thank both speakers for the very nice work. I would like to thank all the members for having connected. And please, if you have any ideas, please don't hesitate to contact, it says it here on the screen, Louise Colville, for ideas of speakers and subjects. And thank you to everybody, and have a nice holidays for the winter.
Yeah, thank you also from my side again. And this webinar was recorded and can be watched on the ISTA YouTube channel in a few days' time. And probably there will be a link also from the ISSS website to this at the same time. Thank you very much, and enjoy the holidays. Thanks and take care. Bye-bye.
Bye-bye.